Hi everybody, we are here to talk about meiosis. Meiosis occurs in eukaryotic organisms that reproduce sexually. So this includes plants and animals. Meiosis is a two-part cell division process that produces sex cells with one-half the number of chromosomes as the parent cell. There are two phases of meiosis, meiosis 1 and meiosis 2. At the end of the mitotic process, 4 daughter cells are produced.
Before a dividing cell enters meiosis, it undergoes a period of growth called interphase. The G1 phase is the period prior to the synthesis of DNA. In this phase, the cell increases its mass in preparation for cell division. The G in G1 means gap, and the 1 means first. So the G1 phase is the first gap phase. S phase. The period during which DNA is synthesized. In most cells, there is a narrow window of time during which DNA is synthesized. The S means synthesized. G2 phase. The period after DNA synthesis has occurred but prior to the start of prophase. The cell synthesizes proteins and continues to increase in size. The cell still has nucleoli present in the later part of interphase.
During prophase 1, chromosomes condense and attach to the nuclear envelope. Synapse occurs. A pair of homologous chromosomes lines up closely together, and a tetrad is formed. Each tetrad is composed of 4 chromatids. Genetic recombination via crossing over may occur. Chromosomes thicken and detach from the nuclear envelope, similar to mitosis. The centrioles migrate away from one another in both the nuclear envelope and
Centrioles?
I messed up right there so bad. I was done.
Alright, continue.
I'm done.
Continue.
Mike, you gotta make sure you read it before you say it.
I didn't have time to read it. You both had the script.
Don't stop believing. Continue.
Okay, I'm restarting at centrioles.
Centrioles.
The centrioles migrate away from one another, and both the nuclear envelope and nuclei break down. The chromosomes begin migration to the metaphase plate.
During metaphase, tetrads align tetrads align at the metaphase plate.
Hey, start over.
During metaphase, tetrads align at the metaphase plate. The centromeres of homologous chromosomes are oriented toward the opposite cell poles. During anaphase, chromosomes move to the opposite cell poles. Similar to mitosis, microtubules such as kinetochore fibers interact to pull the chromosomes to the cell poles. Unlike in mitosis, sister chromatids remain together after the homologous chromosomes move to opposite poles.
During telophase, the spindle fibers continue to move the What is that word?
Homologous?
Homologous chromosomes to the poles. Once movement is complete, each pole has a haploid number of chromosomes. In most cases, cytokinesis, division of the cytoplasm, occurs at the same time as telophase 1. At the end of telophase 1 in cytokinesis, 2 daughter cells are produced, each with one half the number of chromosomes of the original parent cell. Depending on the kind of cell, various processes occur in preparation for mitosis too. However, there is a constant. At the end of telophase 1, the genetic material does not replicate again. The cell enters into prophase 2.
During prophase 2, nuclear membrane and nuclei break up, while the spindle network appears. Chromosomes do not replicate any further in this phase of meiosis. The chromosomes begin migrating to the metaphase 2 plate at the cells you created here. That's you. Again. During metaphase 2.
Can you speak a little louder, please?
During metaphase 2, the chromosomes line up at the metaphase 2 plate.
Oh shit.
At the metaphase 2 plate.
I got no way. I'm restarting because you.
Okay.
No, just talk like you're
During
talking to
metaphase
teacher.
2, the chromosomes line up at the metaphase 2 plate at the cell's center. The
Connect-core?
kinetochore
Connect-core.
fibers of the sisters chromatids point towards opposite poles.
During anaphase 2, sister chromatids separate and begin moving to opposite ends or poles of the cell. Spindle fibers not connected to chromatids lengthen and elongate the cell. Once the paired sister chromatids separate from one another, each is considered a full chromosome. They are referred to as daughter chromosomes. In preparation for the next stage of meiosis, the two poles also move further apart during the course of anaphase 2. At the end of anaphase 2, each pole contains a compilation of chromosomes.
During telophase 2, distinct nuclei form at the opposite poles. Cytokinesis, division of the cytoplasm, and the formation of two distinct cells occur.
The final result of meiosis is the production of 4 daughter cells. These cells have 1/2 the number of chromosomes as the original cell. Only sex cells are produced by meiosis. Other cell types are produced by mitosis. When sex cells unite during fertilization, these haploid cells become a diploid cell. Diploid cells have the full complement of homologous chromosomes.
Yay! But what can go wrong?
Non-disjunction, that's what can go wrong. Disjunction normally occurs during the
You got this.
during the anaphase of mitosis and meiosis. During the anaphase of mitosis, sister chromatids separate and move towards opposite poles of the cell. In meiosis, disjunction happens when homologous chromosomes move apart towards the opposite poles of the cell in anaphase. Non-disjunction is failure of homologous chromosomes or sister chromatids to separate properly during cell division. And these are our sources.